Hello, I'm Lauren Puryear, and I'm a clinical genetic counselor at the University of Washington in Seattle, Washington, as well as a clinical assistant professor for the UW Genetic Counseling Graduate Program. I'm very glad to be here today to talk about the genetics and molecular biology of vascular EDS. I have no disclosures. As I'm sure you all know, vascular EDS is a condition that causes significant risk for vascular complications and hollow organ rupture. It's hard to get an accurate estimate of prevalence since vascular EDS is underdiagnosed, but it may affect as many as 1 in 50,000 people. It's caused by pathogenic variants in the COL3A1 gene. And I'd like to start with a quick refresher on autosomal dominant inheritance. We all typically have 2 copies or alleles of the COL3A1 gene, one inherited from each parent. In this diagram, the mother is shaded in, indicating that she is affected with vascular EDS. The circles representing her 2 copies or alleles of the gene show that one allele has a pathogenic variant that causes vascular EDS, and the other allele does not have a pathogenic variant. So it has typical DNA sequence for the gene. In this example, dad has 2 typical copies of the gene, so he does not have vascular EDS. And when he fathers a child, he will either pass on one allele or the other. Mom will also pass on one allele or the other. So each child has a 50% chance to get the allele with the pathogenic variant and a 50% chance to get the allele without a pathogenic variant from her. About half of people with vascular EDS inherited a pathogenic variant from a parent with the condition. The other half of people with vascular EDS have it as the result of a de novo or new mutation. We all have a few new mutations that happened in just the sperm cell or the egg cell that created us. This helps make evolution possible, but it also makes some people the first person in their family to have a genetic condition like vascular EDS. This affected person now has the pathogenic variant in all of their cells, including their gametes, so they have a 50% chance to pass it on each time they have a child. There are a few rare families in which gonadal mosaicism has been identified, when 2 parents who don't have vascular EDS have multiple children who are affected and have the same pathogenic variant as each other. This occurs when a mutation happens during embryonic development in one of the parents. So in this example, in probably one sperm progenitor cell in dad. So he produces a small population of sperm cells with a pathogenic variant, even though he doesn't have the condition. For this reason, we offer genetic testing to all siblings of an affected person, even if both parents test negative using blood or saliva. This diagram shows the COL3A1 gene. It's located on the long arm of chromosome 2. And has 51 exons or protein coding segments of DNA. In this diagram, the exons are all shown nose to tail, but in the actual gene, they're separated by introns or segments of DNA that are not part of the protein coding sequence. The light yellow bar shows the portion of the gene that encodes the triple helical domain, which is the part of the protein that actually ends up in a type 3 collagen fibril. The rest of the gene encodes material that's important for assembling the triple helix and getting it to where it needs to be, but will eventually be cleaved off of the triple helix. This may look familiar from high school or college biology. If a person has a pathogenic variant in COL3A1, that's in the DNA, in the nucleus, the instructions in that gene are copied or transcribed into a segment of messenger RNA. And these mRNA molecules undergo some processing to remove the introns, and then leave the nucleus and find their way to ribosomes in the cytoplasm of the cell. Ribosomes translate the mRNA or read the instructions and assemble the amino acids in the appropriate order to create a protein. There's a pattern to naming collagen genes, as you see in these examples. COL1A1 encodes type 1 collagen alpha 1 chain. COL1A2 encodes type 1 collagen alpha 2 chain. And so the gene we're talking about today, COL3A1, encodes type 3 collagen alpha 1 chain. Unlike type 1 collagen, which combines two different types of chains into its triple helix, type 3 collagen is a homotrimer, meaning that all three chains that make up the triple helix are the same and are encoded by one gene. Here's a diagram showing collagen assembly. Once those alpha-1 chains are produced, three of them are grouped together and wound tightly into a triple helix. The extra material on either end of the helix is eventually cleaved off, and the molecules are assembled into collagen fibrils. So how does a pathogenic variant in COL3A1 disrupt this process? If we zoom in and look at the triple helical domain, each individual polypeptide chain is twisted, and then the three chains twist together. The chains can fit very tightly together because every third amino acid in each chain is a glycine, which is the smallest amino acid. So as you can see in the cross-section shown here, the glycines meet in the middle of the triple helix. The sequence of amino acids in the triple helix of collagens is often referred to as glycine XY. The amino acids in the X and Y positions can vary, but the glycine is consistent in every third position. This is a busy graphic showing the structure of amino acids. The thing I want to draw your attention to is that glycine in the upper left corner doesn't have a side chain, just a tiny little hydrogen highlighted in green. All of the other amino acids have stuff sticking out in that same position. So substitution of any of them for glycine disrupts that tightly wound helix. Many pathogenic variants in COL3A1 are substitutions of another amino acid for a glycine in that triple helical domain. Other types of variants in COL3A1 can also cause vascular EDS. You may hear people talk about null variants, which basically means a genetic variant that creates a stop message partway through the gene. So the RNA transcript doesn't include the whole sequence of the gene and is significantly shortened. These shortened transcripts are usually unstable, and the cell degrades them before they can be used in translation. So no protein is actually made from that copy or that allele of COL3A1. The other variant type shown here is a splice variant. So as it sounds like, it's a variant that disrupts splicing, that processing of the RNA transcript to cut out the introns of the gene and leave just the exons, the segments that encode the protein. Some splice variants lead to exon skipping, in which one or more exons get left out of the mature RNA transcript. Other splice variants may lead to inclusion of introns that should not be in the transcript. And some splice variants may lead to a very shortened transcript that is unstable and is degraded by the cell. So essentially like a null variant, and no protein is produced from that allele. There's not always a way to tell what the outcome of a splice variant will be. And so in some situations, we can use RNA analysis to give more information about what happens with splicing. The consequence of having a null variant is that no protein is produced from the instructions in that allele of COL3A1, but normal protein is still produced using the DNA instructions in the other allele. So the person makes normal type 3 collagen, but less of it than a person who has two typical copies of COL3A1. For a person who produces abnormal alpha-1 chains from one allele, they're also producing a similar number of normal alpha-1 chains from the other allele, but they don't end up with half normal type 3 collagen and half abnormal type 3 collagen molecules because the chains are randomly combined into groups of 3 to create that triple helix. So if you look at the possible permutations of combining abnormal and normal chains in randomly selected groups of 3, 7 out of 8 groups are going to include at least one abnormal chain. This difference is why, on average, people with null variants have fewer vascular events and later age of onset than people with variants that produce abnormal protein. So genetic information for a patient can be useful to give some information about the natural history for the type of variant they have. And in some cases, it's very useful for folks who are thinking about reproductive planning. And I think someone will talk more about that in a different talk. These are the key things I think it's useful to remember about the genetics and molecular biology of vascular EDS. Pathogenic variants in COL3A1 interfere with the structural integrity of type 3 collagen or cause less type 3 collagen to be produced. And it's always important to remember the inheritance pattern of this condition as you work with patients, and especially as you work with families. That's it for me. I would like to thank the EDS Society for hosting this very important event. And as always, thank you to Peter Byers and Shireen Shalhoub and all of the patients and families that I work with for everything that you've taught me.